A tumor cannot grow beyond the very small size without blood supply. As it grows, its cells need oxygen and nutrients. But diffusion alone is not enough. As tumor triggers the formation of new blood vessels, these blood vessels supply it with oxygen and nutrients, allowing it to continue growing and potentially become much larger. Now, the important question is, how does the tumor actually create its own blood supply? To understand this, we need to look at what happens inside a growing tumor. The tumor begins to outgrow its blood supply. When a tumor is very small, its cells can receive oxygen and nutrients from nearby blood vessels. Oxygen can move through the tissue by diffusion, but this process only works over a relatively short distance. As the tumor gets larger, some of its cells become too far away from the nearest blood vessels. The result is a shortage of oxygen. This low oxygen condition is called hypoxia, and hypoxia is one of the major signals that can trigger angiogenesis. The tumor senses the lack of oxygen. Tumor cells have molecular systems that respond to changes in oxygen levels. One of the most important regulators is a group of proteins called hypoxia-inducible factors. Under normal oxygen condition, a protein called HIF-1 alpha is continuously broken down. But when oxygen levels fall, this protein becomes more stable and accumulates inside the cell. It can then interact with another protein. Together, they can activate specific genes involved in the response to low oxygen. One of the most important products of this response is VEGF. The VEGF becomes a signal for new blood vessels. This protein stands for vascular endothelial growth factor. The tumor and other cells in the tumor microenvironment can release VEGF into the surrounding tissue. This protein then travels toward nearby blood vessels. You can think of it as a molecular signal that tells the surrounding vascular system there's an area here that needs a blood supply. This protein also activates endothelial cells. Blood vessels are lined by specialized cells called endothelial cells. These cells have receptors on their surface that can detect VEGF. One particularly important receptor is called VEGFR2. When this protein binds to its receptor, it activates signaling pathways inside the endothelial cells. These signals change the behavior of the cell. The endothelial cell becomes more likely to survive, divide, and migrate toward the source of the signal. The existing blood vessels prepares to grow. Before a new vessel can form, the surrounding extracellular matrix has to be remodeled. Activated endothelial cells and nearby cells release enzymes that modify and break down parts of the matrix and the vessel surrounding structures. This creates space for endothelial cells to move out from the existing vessels. Endothelial cells begin to migrate. Now some endothelial cells start moving toward the tumor. They don't move randomly. They respond to chemical signals in their environment, including guardians of VEGF. This process is called chemotaxis. One group of cells takes the lead and forms a structure called tip cell. Behind it are stalk cells, which proliferate and help extend the developing vessels. The vessels start to sprout. The tip cells move forward through the surrounding tissue. The stalk cells behind it divide and follow. Together, they create a growing extension from the original blood vessels. This is called a sprouting angiogenesis. At this point, it is not yet a fully functional blood vessel. It is more like a growing cellular tube that is gradually organizing itself. A lumen begins to form. As endothelial cells organize, they create a central hollow space called a lumen. This lumen is essential because it will eventually allow blood to flow through the new vessel. The developing vessel can also connect with another vessel. This connection is called anastomosis. Once the network becomes connected, blood can begin flowing through the newly formed vessel. The new vessel becomes stabilized. A normal blood vessel doesn't consist only of endothelial cells.
It also needs supporting cells and extracellular structures to stabilize it. One important type of supporting cell is called parasite. During normal vascular development, parasites help strengthen and stabilize blood vessels. However, tumor blood vessels often have abnormal or incomplete support. That's one reason tumor vessels can be fragile and unusually leaky. The tumor now has access to circulation. Once new blood vessels reach the tumor, they can provide oxygen and nutrients. This can allow tumor cells that were previously limited by oxygen and nutrients availability to survive and continue proliferating. But there's another important consequences. Because tumor blood vessels are often abnormal and leaky, they can also make it easier for some cancer cells to enter the circulation. If those cancer cells survive in the bloodstream and establish themselves in another organ, this can contribute to metastasis, or the spread of cancer to distant part of the body. Angiogenesis is not simply the tumor getting more blood. It is a complex biological process involving low oxygen, HIF protein, VEGF signaling, endothelial cells, extracellular matrix remodeling, cell migration, proliferation, vessel supporting, lumen formation, and vascular stabilization. And that's why angiogenesis is considered an important part of tumor progression. The tumor doesn't just grow larger, it changes its environment to encourage the body to build new blood vessels that can support that growth.
The biology behind cancer is incredibly complex, but understanding this process helps us to understand how a tumor or a cancer actually grows and spreads. And let me know in the comments if you have experienced cancer. Maybe your experience can help somebody else.